Hello everyone, in this video I'm going to be talking about gene markers and a process known as replica plating. Now this is all part of a larger topic in transforming vectors. When we look at DNA technology and genetic engineering, we think about how we transform a vector or a carrier of genetic information as part of that process. And usually we think about the plasmid, the ring of DNA in a bacteria, as being our vector. So what I'm going to do in this video is talk about how we can use gene markers and a technique known as replica plating to see that we have indeed transformed that vector. So let's start with the basics, because we need to first of all understand why we're going to do what we're going to do. So here is our ring of DNA. A circular ring of DNA in a bacteria, and this is our plasmid. Now we need to put that plasmid into, ultimately, an empty bacterial cell, because we would have extracted that plasmid from the bacteria originally and altered it in such a way, or transformed it, so that we would have inserted new DNA into that plasmid. So we would have made recombinant DNA. So what I'm going to have here is a section of that plasmid, let's say that we've inserted, we've actively put into that ring as part of our genetic engineering process. Now, ideally, what we're looking for is, if I just draw a very rough sketch, is for that plasmid to be incorporated into the bacteria. So we're looking for that bacteria, antibacterial cell. I'm just going to colour in just for completeness sake, very roughly. We're looking for that plasmid to get into that bacterial cell. Now, for that to happen, we're going to require a slight temperature change. So I'm going to put temp change, because we're going to need to slightly increase the temperature, and also we're going to need calcium ions. Now, calcium increases the permeability of the bacteria, allowing them to take up the plasmid, but this only happens in 1% of the bacteria. So we're only looking at a success rate of 1%. Some don't take it up, so some bacteria don't take up that plasmid, and some plasmids actually close up without the DNA fragment in place. So our plasmid closes before our new DNA even gets a chance to be inserted into it. So that's where this video is really picking up from. So when DNA fragments are inserted into existing gene segments of a plasmid, we say it transforms it, and it usually alters its function. Now those sections of the plasmid act as what are called gene markers, and they allow us to see if the plasmid has properly incorporated the fragment before closing up, and if that bacteria has taken it up. So first of all, let's think about antibiotic resistant markers. Antibiotic resistance markers, so in fact we're going to list the type of markers we have here. So number one, the one that I'm going to talk about first, are the antibiotic resistance markers. Now, these are less commonly used than the two other markers I'm going to refer to, the fluorescent markers and enzyme markers. But it's a really good process, if you like, to talk about, because it ultimately explains how quite simply we can use these markers to check what we've been doing. So these antibiotic resistance markers we find on our plasmid. So if we just put a plasmid in place here, what we find is that certain plasmids have two genes on them for resistance. One of those is a gene for ampicillin antibiotic resistance. And there's another gene for what's called tetracycline resistance. So two antibiotics that can destroy this bacteria are ampicillin and tetracycline. And we find that certain plasmids have genes allowing them to be resistant to these particular drugs. So with these genes, our bacteria is able to break down ampicillin or break down tetracycline and survive. Now what we can do is ultimately cut one of those genes with our restriction enzyme and insert the gene that we are studying into that particular segment. So what we're going to imagine, if you like, let's say for the sake of argument, here is our, just going to call it gene, the gene that we want to insert into our plasmid, our vector. And I have done a whole video on genetic engineering to explain this in more detail, but let's say that we've cut open that vector here. So we've cut open this plasmid, this ring, and we've inserted our gene into there. So it looks a little bit like this. This is what we're imagining it now looks like. We have ampicillin. We've got our gene. And this, if you like, I mean, I've not drawn it to scale, but where I've got the green, that was the tetracycline gene, but we have altered it because we've inserted our own purple gene. Now this purple gene could be a section of DNA coding for insulin, let's say for example. We've inserted our own gene into that region where we have a base sequence coding for tetracycline resistance. Now when we do that, we alter that particular gene. In fact, I don't want to use the word destroy it, but we stop it from functioning. So by transforming or changing that tetracycline gene, we stop it from working. So we're going to use this idea and talk about the process of replica plating. So for this, we're just going to, as I normally do, shrink the screen just a little bit. Now I want you to imagine that we have a petri dish over here, and in a culture medium I have cultured colonies of bacteria that I have hopefully transformed. In fact, the proper word to use is made transgenic. A transgenic organism is one where the bacteria has been altered, where I have DNA from two sources. So what I have is transgenic bacteria, I'm hoping for, cultured on this dish. So I'm just going to draw some colonies of bacteria, and I'm deliberately using different colours just to make a particular point. Let's see in one moment why. So let's say we have a dish, petri dish, and I'm culturing some bacteria. Now what I do, the first stage in replica plating, is to take a dish, and in that dish I have some ampicillin. So I have a whole petri dish of ampicillin. And I'm going to grow these bacteria, I'm going to culture them in that dish of ampicillin. Now I want you to imagine that this is the result of what I see when I do that. So you notice the red, pink, green and blue dots are thriving, so they're growing on ampicillin. But you'll notice that the yellow dot here and the dark blue dot here are missing. Now straight away we can say that if our plasmid was properly taken up into the bacteria, and we're imagining that we transformed the tetracycline gene, but we left intact the ampicillin one, the ampicillin resistant gene, those that have survived on this ampicillin plate must have the gene for ampicillin resistance. Otherwise, in a plate full of ampicillin drug, they would be killed. So we can see that the two, where I've put an X, the two bacteria colonies, the one in dark blue and the one in yellow, have been destroyed in the ampicillin, so they don't have ampicillin resistance. So they mustn't have that plasmid that has the ampicillin resistance gene. Now, what we're going to do is make what's called a replica plate. So we're going to take a plate, but in this plate, we're going to put in tetracycline drug. So we now have tetracycline drug, and it's called replica plating because we are literally, if you like, plating this as a replica of what we've got already. So we're going to take a sample of the colonies on this ampicillin plate, and using sterile techniques and aseptic techniques, we're going to transfer exactly into the same position these colonies on the tetracycline. Now let's imagine that we transfer, so let's say with the red for example, so what I'm going to do is transfer this over to here. I want to imagine that I've transferred the red, the pink, the green and the blue. But when I allow those colonies to grow, I only have a red and a green dot remaining. So if you like, my pink bacterial colony and my blue bacterial colony have died. Now, if we have correctly transformed that plasmid, if we have managed to properly insert our purple gene, so our insulin gene, let's say, into where the tetracycline resistant gene is, then we should stop that tetracycline gene for resistance from working. We should stop it working completely. It shouldn't function. So that plasmid would lose the tetracycline resistance ability. Now what we can see is that our pink and light blue dots, they're not growing on the tetracycline plate. They have been destroyed by tetracycline. Now it's those two that we actually want. They're the ones we wanted, because that has shown us that not only have they survived the ampicillin plate, which means they've taken up the plasmid in the first instance because they have the ampicillin resistant gene, but it's in the blue and the pink bacterial colony that we've actually managed to correctly transform our gene, our tetracycline resistant gene. Because if the process did work, then technically we should have stopped that resistant gene from working. The red and the green dot, so this red and this green colony here, the fact that they can survive on tetracycline drug implies that this tetracycline, and I'll highlight it in blue, it shows that this here, this tetracycline gene hasn't been cut properly. Or maybe the plasmid has closed up and our gene hasn't been inserted properly, because for whatever reason that tetracycline resistant gene is still operating. If it wasn't, then our gene that we wanted to insert would have gone in and stopped it from working. So the benefit of replica plating is that now we can see which particular plasmids have been taken up and properly transformed. We can go back to our ampicillin plate and culture just the pink colony and the blue one. Because if we look at this tetracycline, this tetracycline plate, the ones we're after have actually been killed. But that's why it's a replica copy. So we can go back to our ampicillin plate and know that this pink and blue colony, not only have they taken up the plasmid because they've survived on ampicillin, so they have the ampicillin resistant gene, but we know that tetracycline kills them. So those two also have a non-functioning tetracycline resistant gene. So we have successfully transformed them. We've managed to put our gene into those slots, if you like. And that is replica plating. Now there are two other types of gene markers that I said. Fluorescent markers. Now fluorescent markers are a lot simpler to use. I'll just shrink this again just to make a note, just to write down here. So we've got fluorescent markers. Now fluorescent markers use what's called the green fluorescing protein. I mean, this is the best example that I'm giving here. Jellyfish have what's called GFP, green fluorescing protein. And that can be transformed when DNA fragments are inserted into it. So we're going to imagine that we've got our plasmid originally. This in red is the gene that we want to incorporate into the plasmid, but we're going to imagine, and again I'll just colour it in another shade of green to represent this here, that I'm colouring in green, is our green fluorescing protein gene. So if a plasmid has been successfully transformed, the bacteria shouldn't produce green fluorescent protein, so they won't glow. So we can use microscopy to detect such cells. So this green gene, if you like, normally when it's present would produce green fluorescent protein, and this whole bacteria would glow, if you like. But if we have successfully transformed the plasmid and we've put in our red gene and it has worked, the plasmid hasn't closed up yet, it's accepted our red gene, then we should have stopped the green fluorescing protein gene from working, so it shouldn't glow. And there is a third. I'll just separate these two from the ones above. There is a third type of marker, and these are the enzyme markers, and they work by the same principle. There is a gene for lactase enzyme. Now that lactase enzyme is able to turn a colourless substrate. Now you don't need the name of it, but it's called X-gal, X-gal, but for this we'll just say it's a colourless substrate. It turns it blue. So when lactase enzyme, or when the lactase gene is functioning and produces lactase enzyme for the bacteria, it's able to turn a colourless substrate blue. Now bacteria which have transformed the lactase gene will grow colonies that won't cause a colour change. So if we had a plasmid and we inserted the gene that would code for lactase, and then another gene, the one that we're actually interested in, if our gene has successfully been taken up by the plasmid, then the lactase gene will have been transformed, and we shouldn't be able to get that lactase gene property. We shouldn't be able to turn the colourless substrate blue, because we won't have lactase enzyme being made. If it hasn't worked though, we use this enzyme marker to detect that. If it hasn't worked, if we've not transformed the plasmid, then the lactase gene will be intact. It will code for lactase enzyme, and that colourless substrate will turn blue. So this video is just a little bit about transforming vectors, gene markers, and replica plating. It's a bit longer than normal, but it's because it's a bit of an in-depth topic. Okay, hope all that helps.